Hello. Good morning.
Hello.
Good morning, Professor Naqvi. Yeah, we can hear you.
Sure. It is good afternoon for me.
Oh, yeah. It's for you. Good afternoon.
But can you hear me clearly? Or
Yeah,
sure.
we can hear you. Maybe you can just speak a little bit louder, like during the presentation.
Sure. Yeah. Is
Yeah.
this clear? Or?
Yeah. Now it is better.
Okay.
So, Professor Naqvi, we will be starting in 1 minute.
Sure. Are you able to see the slides? Or?
Yes. Can you just go to the play mode, that slideshow mode?
Is that better?
Yes. Now it is good. Okay.
So good morning, everybody. And it's a pleasure to present our recent work. And I thank Abhishek and Nazmiara for this opportunity to talk to you about some key challenges in both traditional and urban agriculture.
Okay.
These two.
Sorry for interrupting. So just give me 1 minute. I just introduce you with our audience. So then you can start. Okay, good morning and welcome back to the session. So we are extremely happy to have Professor Naqvi among us today. He obtained his PhD from Maharaja Sayuraja University of Baroda and the International Rice Research Institute, Philippines. Subsequently, he worked as a Rockefeller Foundation Fellow at IRRI and later moved to the Institute of Molecular Agrobiology, Singapore, for his postdoctoral work in eukaryotic cell division. Dr. Naweed joined the Insect Life Science Laboratory as a senior scientist in 2002 and served as an adjunct professor at the National University of Singapore. He has more than 50 internationally acclaimed research publications. He has a diverse research interests, including infection-related morphogenesis in fungi, surface sensing and signaling networks underlying fungal pathogenesis, chemical biology of host-pathogen interaction, and molecular patterns and natural variation in the blast pathosystem. With this, we welcome Professor Naweed to deliver his talk entitled Harnessing Plant Microbe Systems for Precision Agriculture. And I have a small announcement to our participants. So after the talk, after the session, please go to the link with the YouTube. And if you click there, you can get the certificate after the talk. Thank you. So please welcome Professor Naweed to start his talk. Thank you, Professor Naqvi. You can start.
Thank you very much for the introduction and welcome back to what I was saying earlier. So the two key challenges that we have as our goals, which are challenges and opportunities both for traditional agriculture as well as urban agriculture, as far as Singapore is concerned, are to improve yield and resilience in food crops, and more importantly for us to develop novel traits. And in the context of the present talk, I think I would refer to this as more of engineering immunity in a very precise manner, or precision immunity as we refer to it. So to address these two questions, what we do is to harness the highly intricate tripartite interaction between host microbe and the environment to precisely engineer new traits that would allow us now to look at both the growth and resilience and improve them in a very controlled manner. And our model system for such studies is well known to most of you, since Nazmir also works on the same disease, which is the rice blast disease. And we are particularly interested in this window, which I've highlighted in red here, which is the very critical transition that the blast fungus makes once it enters the plant. And this transition refers to its move from keeping the host cell alive, which is biotrophy, to really rampant killing that it is notorious for, which is necrotrophy. So this transition window, biotrophy to necrotrophy, is very crucial for the elaboration of the devastating disease symptoms that Magnaputa oryzae or the blast fungus is well known for. Now, the questions that we have, at least in this window, is to really understand the metabolic interactions that occur between the blast fungus, Magnaputa, and its host plants, which of recent have also included wheat to a great extent, given the recent epidemic in Bangladesh. So the key question that we would like to address here is, how does Magnaputa try to keep the host alive by suppressing the host defense during this very critical window of the biotrophy to necrotrophy transition? And we also address this issue by looking at the main gateway or the interface, which I've highlighted here in red. This is the biotrophy interfacial complex as identified or established initially by the Valent lab. And this basically is the conduit for the transport of avirulence factors, effectors, and small molecules from the fungus into the host in that window that I was referring to, which is the biotrophy to necrotrophy transition. So the key interest that we have here is to basically deconstruct this interaction and, more importantly, identify the metabolic regulators of both the plant immunity and the fungal pathogenesis in this very specific window. And we want to achieve these goals in a highly controlled manner, because from our earlier work, including our collaboration, which is shown here in the middle, which is in Arabidopsis, what has been seen is that if you shift the balance too much towards the defense, like having very rampant necrosis occurring, or disease resistance, which is also leading to a deficit in growth, then you have a lot of necrosis happening. So the entire metabolic resource is now getting shifted to be used in defense, and this is always at the cost of growth. So you have stunted growth or reduced growth, but highly robust defense in this case. So what we would like to do is to keep this balanced in a way that we have somewhere in the middle, where we have robust growth as well as equally important precise defense mechanism. So you can imagine our excitement when we found a mutant in the blast fungus, which actually induced this defense in a very controlled manner. And this is something really novel, and that we had never, or neither anyone in the community has seen before. So on the top, what you see is the wild type fungus that is colonizing the first invaded cell. So the black shadow that you see with an asterisk is actually the infection structure of this fungus that would allow it to enter it into the first epidermal cell. But it, like I mentioned earlier, keeps the host defense suppressed so that it can be a biotroph and try and regain its metabolic potential to become necrotrophic later on, or in the next one day or so. On the bottom panel, what you see is this mutant that, although enters the plant like the wild type, but does not live for more than 24 hours. So if you look carefully, you can see that the ghost of the hypha is there, and this has been completely obliterated because of this very novel defense that has been mounted against this mutant fungus. And this defense is very novel in the sense that it is highly specific only to rice as the host. It is not seen in barley or in wheat against this mutant. And what it also includes is the anchoring of these very specific inclusion bodies that seem to somehow home in to the hyphae and then very specifically killing it in a very, very short time. So we were very curious about this, and we basically took two very distinct approaches to look at it. One was cell biology, and the other one was very specific detailed metabolomics supported by very high-end tandem mass spec analysis to really identify the key regulators of this novel defense response in rice, which is not happening in wheat or barley, like I said. So we have those two as controls side by side to look for exactly the kind of genes that are coming up or the metabolites that are being made in response to rice from both the wild type as well as the mutant ABM. And through comparative metabolomics and the mass spec analysis, we were now able to look at nearly 9,000 chemical signatures in this interaction. And these broadly fall, at least in terms of Magna-Rice interaction, into these four major classes now. So we have a very intricate association of this novel defense pathway with the wounding response. And there are very specific and novel phytoalexins that are being made. And these are ones that we think are responsible for killing of the mutant. There's a subtle association with the redox homeostasis. So ROS does seem to play a role, but not as much as these other metabolites. And we have three absolutely novel metabolites that are responsible for this very specific and novel, robust defense mechanism. And these three metabolites, one of them is jasmonate derivative, which is methylated, so methyl jasmonate. And this is the one that activates that novel defense response. The other one is a hydroxylated derivative of jasmonic acid that is the suppressor of host defense during the biotrophic phase by the fungus. And I should maybe remind you, I'm not going into the details, but these are actually the jasmonate mimics that are being produced by the fungus and not the host. So where we have a unique situation where the fungus has opted as adaptive evolution, where it has now gained the importance of biosynthesis of these phytohormones that are really like the host counterparts. But at the same time, it is also using two of these modified versions now for its own establishment and interaction with the host tissue. So these are the novel metabolite regulators of this novel defense pathway that we identified. And what we have now gone on to do is to also engineer very precise biosensors to monitor both methyl jasmonate and the 12-hydroxy-jasmonate dynamics in real time in live plants. So what I'm showing you is basically the first prototype, which is an example that we established in Arabidopsis roots. And here what you have is a marker for a co-repressor complex. So basically what it simply implies is that if you lose the repressor, then that means that the jasmonic acid signaling in the host is activated or elevated. So what we now know, and we can monitor this in real time, is that the presence of methyl jasmonate, which is an inducer of jasmonic acid signaling, destabilizes this complex very quickly within a matter of minutes. And at the same time, this novel metabolite that is produced by the fungus is the one that actually stabilizes it, and basically the jasmonate signaling is now dampened. And that is the reason why the fungus can suppress the host defense using this novel metabolite. We've also looked at the cell biology of this interaction. So this is all unpublished data. And what we were very keen on looking at is the intracellular or subcellular trafficking of these novel metabolite effectors, including the enzyme that makes these, ABM, which is resident in this interfacial complex shown in red in the schematic. So what we found recently was a very strong association with the novel vacuolar compartment that also includes some bits of the late endosomes as well fused to them. So the Hobbs complex and the endolysosomal compartment are strongly associated with the blast biotrophy interfacial complex in red. And you also have these very specific interaction points where you have vacuolar components or some of these latent endolysosomal vesicles trafficking into the BIC or the interfacial complex from these vacuoles. So this is a rather new finding because we never really thought that the vacuole would be so important for delivery of some of these effector molecules. And at the same time, if you can look to the image on the right, which shows the retromer. So what we found subsequently through some mutant analysis was that the retromer is the one that is actually carrying some of these cargo, as some of these metabolites as cargo, and is getting also trafficked in these cases. So what is also interesting is that if you disrupt the RAB7 or the late endosomal fusion machinery, then you have not just the stoppage of this kind of signaling, but at the same time you also disrupt the signaling, the setting up of the interface itself in these cell types.
Professor Naqvi, just
Yeah.
I just want to mention one thing, that for the convenience of the speakers, bell will be there at 3 minutes prior to the end of the talk, your talk.
Sure.
Okay, thank you.
My presentation seems to have stuck. I'm trying to get over it. Just give me a minute.
Yeah, sure. Oh, there is some problem with his internet. Okay, let's see. Yeah, I hope he'll be back soon.
Anybody has his number to contact him? Again, he may not realize that he has disconnected.
No, he told me that time that his presentation suddenly stopped. So there must be some kind of network issue. Yes, so you are here, Professor Naqvi.
Hello.
Yeah, yeah, we can hear you.
Hello.
Yes, we can hear you.
Are you able to see the slides now? Or?
Yeah, yeah.
Sure.
So you
Sorry
can.
for the interruption. I
No,
don't
no,
know.
no, not a problem. This is the virtual thing, and these things happen. You may proceed.
So what I was saying is that when you lose the RAP7 signaling, then you also lose the ability of the fungus to establish this conduit or the interface in a proper manner. So on the image in the right, you have, unlike the wild type where you have a very specific, very specified interface, here you have the interface all over the place. So you have disrupted interface in just about all over the membrane targeting with the host. So what we are trying to achieve with this kind of approach is to have a precision engineering of this novel defense pathway in rice, and hopefully to also take it to barley and wheat. And like I mentioned earlier, one of the key specific points for the specificity of this novel defense is that it is triggered by methyl jasmonate that is provided, well, it is produced by the fungus itself. And it has a very strong correlation with the bonding system, and is triggered by the bonding system. And we have very strong association with these antifungal activities, especially those phytoalexins and these inclusion bodies that seem to know where the invading fungus is and to kill it. Now, so far I've been telling you about our work on the pathogenesis part, or the interaction between a pathogen and the host. And I would switch gears and tell you more about how we look at the host microbe interactions in the beneficial scenario for improved crop production, and in this case, for mostly targeted at urban cultivation in Singapore. So the tripartite interaction between microbes, nutrients, and the defense is something that is still at the back of our minds, where we're looking for very specific microbiome constituents to effectively improve production and enhance the nutritional benefits of these green leafy vegetables that are cultivated in Singapore. So this is a fairly large project with collaborations from the National University of Singapore and the Environmental Research Institute, and one of the biggest vegetable producers here, which is the Biotechnology farm. And what we are doing here is to engineer microbiomes of known composition to enhance yield and resilience in the locally grown vegetable crops. The first part was to identify the composition by using both cultivation methods as well as metagenomics, and then the functional part to identify the beneficial metabolites that would enhance this growth. And we also want to develop disease resistance using these microbiome constituents, especially against the wilt disease, which is very common here. So what was exciting was some of these metagenomic analysis and the diversity index that we found. So what we're looking at are these three layers, which is the bulk soil, the rhizosphere, the rhizoplane, and the endosphere, to look at what are the microbiome constituents that are specifically associated with these three compartments in the rhizosphere. And what we found was a very interesting diversity in terms of looking at about 2,000 OTUs or species that are very highly specific in each of these individual compartments. And what is most interesting and functionally very relevant and important for us is that when you provide this cultured microbiome, to the roots in these highly quantitative assays, then you see a very clear benefit in both in terms of growth promotion and in terms of resilience. So on the left, you see the functional effect of these microbiota when provided to these seedlings that are growing on lab medium. Under soil conditions, it stays the same. So even when you provide them in soil, they do promote growth significantly. So up to 35% increase in the biomass, typically, both for two different types of vegetables, scallion and jasmine. And what we've gone on to do is exactly what we did for the blast system, which is to look at these interactions between the microbiome, the beneficial microbiota, or the beneficial fungi, and these vegetable crops by looking at firstly the hierarchical clustering using multi-omic analysis. So where exactly they are, or where they come from, right down to the species level. And then also look at the metabolic constituents that are responsible for providing this growth enhancement and the resilience. So what we have now are very specific post-microbe interfaces. And what is also interesting is that even these beneficial fungi produce very specific phytohormones that are responsible for enhancing this plant growth. And we have established this by high throughput LCMS analysis and also through chemical complementation analysis. So this is an Arabidopsis mutant in gibberellin, and this is completely complemented by the gibberellic acid that is now being produced by one of these beneficial fungi. And some of these associations are novel. So for example, on the left, you see one of these beneficial fungus growing only between the cells of the roots. So it does not harm either of the two neighboring cells or others, but just thrives very specifically only in the interstitial spaces. And the growth benefit is immense, like I was saying, up to 40% increase, even under low nutrient condition. And the same sort of study also identified one of the really interesting endophytic fungus that is beneficial, again, with a significant increase in biomass. And these are the ones that are now being looked at in high detail. So in summary, I've told you about this precision engineering of this novel defense pathway using metabolites that are derived from a pathogenic fungus. And on the other hand, I've told you about the beneficial host microbe interaction for growth promotion and resilience. And even in this case, you have cross-linked phytohormone transport and signaling that is occurring from the fungus, from the beneficial fungus in this case, to the target vegetable. So with that, I would like to thank my lab, particularly Kayu, who did the work on the microbiome, and my student, Liu Inyo, with the help from Rajesh Patkar, who is now in Andhra Pradesh University, who did the work on the jasmonate signaling. And thank you for your attention.
Thank you, Professor Naqvi. It's a wonderful talk. So may I request the moderator of this session to conduct the question answer session?
Thank you very much, Chandramaji. Thank you very much, Professor Naqvi, for this wonderful lecture. Now the time is for taking some questions. So I would like our DLS faculty member, if they have any question, then they can go ahead. Any question from DLS faculty? So if not, then I'm taking questions from our participant, which are present in YouTube live. So first question is from Omlan Banerjee. Hello.
Hi, yeah.
So the first question is, does increasing microbial genetic distance, that is community dissimilarity, increase with phylogenetic distance between their plant host?
That's an interesting question, actually. So what we did, which I didn't show you in the interest of time, is that we have also looked at what specific microbiota associate with a particular plant genus, for example. So the three vegetables that we are working with, Kala and Choisum and Bok choy, we know exactly which specific species are. So it's almost like the plant decides what it wants to keep and what it wants to get rid of in terms of the microbiota constituents. But coming back to your question about the genetic diversity, I think there is a correlation, very strong correlation there between some very closely associated bonds. And what we have not tried to do is to see whether there is a functional relevance or a metabolic relevance to it, and there is. So genera from the microbiota that are specifically involved, say for example, in a particular pathway, I gave you an example of phosphate deficiency. You do have genetic distance or diversity playing a much more important part in those cases. So microbiota genus or genera that are closer in taking part or being functionally relevant to that particular pathway are always associated together.
Thank you, Professor Naqvi. The next question is from Subhashish Indro. Is there any difference occur in microbe and plant interaction when the plant is under various abiotic stress like salinity?
We haven't tested salinity specifically, but we have tested drought. So, and there is a very strong bearing on the microbiota constituent and the composition when the plants are stressed for water. So we have another collaborator in the program who is also looking at the bacterial microbiome, and they are looking at the salt tolerance. But definitely there is actually, because there are studies, for example, from mangrove microbiomes that are associated with the mangroves, and there is a very strong correlation there between very specific genera that are enriched in these microbiome in response to salt stress.
Okay, Dr. Nazmiara Shabnam has a question to us. Dr. Shabnam, please go ahead.
Hello, Professor Naqvi. I'm
Yes.
very happy to have you in today's session. So I have a question regarding the different types of effectors. So how would you differentiate between different effectors when it is delivered into the host cell?
And at the moment, we are only looking at, so I guess it's easier to do with the proteinaceous effectors. So we can tag them, for example. So AVM can be tagged, but for the chemical metabolites, we are still looking at opportunities to chemically tag them with fluorescent markers.
Okay.
But I guess what we would also like to do is to see whether we can have them as mixtures, because obviously in a natural scenario, you don't have the metabolites operating in a singular manner. So they're always like a mixture. So I guess that's where we are trying to do.
Okay. Thank you so much.
You're welcome.
Professor Naqvi, there is another question from Devjani Chanda. Is there any difference in the interaction between host and microbiome due to the difference in their rice varieties?
So in the rice microbiome, we have not really looked at in detail because there are other labs that are doing it. And also rice is not grown in Singapore. So I guess that's something that we are not looking at. But what I can summarize is the work that people have found in rice microbiome. And there is a very strong correlation even there with respect to different cultivars and also in correlating it with different types of soil that it is grown in. So the culture conditions as well as the species as well as the cultivars do have a bearing on the microbiome.
There is another question by Amonita Vishwas. Do abiotic cells affect host plant and microbe interaction?
Actually, it does. So there are quite a few publications on that. So one of them refers to the production of cytokinin by the blast fungus. And there is a very strong correlation to drought tolerance based on that. So when you have the fungus producing less cytokinin itself, then it is more prone to drought, then the plant is more prone to drought. So basically, it compromises drought tolerance when you have less cytokinin being produced by the pathogen. So there's definitely a correlation. I'm not sure whether the other abiotic stresses have been looked at. The earlier question about salt tolerance was, I guess, in a way related to this question.
Okay, there is a question by Nita Day. Can genetic modification be done in crops other than rice to induce the defense mechanism in them?
At the moment, they would still be at the prototype level. I mean, we do have some of these as transgenics, but cultivation-wise, I guess that's far from, I guess, they're not acceptable at the moment as GM crops. Only maybe in the US and Brazil, for example, but not in Singapore. Which is one of the reasons why we focused on using the microbiome, because then you completely avoid the GM technology there. But at some point, we do need to really address that issue, because the engineering of this novel pathway also requires that kind of modification.
There is a question by Anumita Sharkar. Is there any role of ACC deaminase activity of microbes with the stress tolerance of host plant?
So the other collaborator that I was mentioning that was looking at the bacterial microbiome, they have found a correlation, but this is still unpublished and is also with the same vegetables that I was mentioning. So I think ethylene production also definitely plays a role in regulating the necrosis.
Okay, thank you very much. And another question is from Jannatul Firdaus. Is in different rice variety, is there any difference between defense mechanism for them for survive?
That's very interesting, because now we have genome sequences for 3,000 accessions in IRI. And there is definitely a lot of JIVAS experiments and magic populations that are being made now. And there is very clear difference between the genotypes and their defense capacity. So for example, one of the ones that I worked with as a graduate student and a postdoc earlier in IRI was Oryza minuta, which is one of the wild rice varieties. It's not cultivated at all because it's a very short staged plant, hardly grows much. But it falls in this category where it is really strong defense, but completely compromised for growth. So there are similar different associations. And it also refers again back to the presence of the resistant genes in these various different accessions or cultivars. So what I didn't talk about is the R-AVR kind of interaction that we are also looking at in the lab, especially for AI9 based broad spectrum blast resistance. So there's definitely a difference in the defense capacity of different accessions or different varieties.
There is a question by Mosumi Bishas. Are any particular growth regulators or the metabolite necessary for the shift of biotrophic nature to necrotrophic?
There are definitely a lot of these. So I was referring to you to those 9,000 signatures in my talk in the metabolomics analysis, and we do see some of them very clearly associated with that kind of biotrophic transition. But what we also feel is a bit complicated in terms of its interaction is that it differs between barley, wheat, and rice. So we're not sure whether we are really looking at host specificity or whether we are looking at specificity for the biotrophy to necrotrophy transition. That's something that we are actively looking at.
This question by Rahul, Ms. M. Chatterjee. What plays the most important role in determining if a particular organism will establish as a pathogen or will become a symbiotic in a plant?
I think in general, there is this belief that the pathogens actually evolved from symbionts or the saprophytes. But again, the diversity is so huge that it's very difficult to generalize that kind of system. But there are experiments done specifically by several labs, including from Barry Scott in New Zealand, where they have shown that if you just deregulate the reactive oxygen species production in the fungus, in a symbiotic fungus, then it actually becomes more like a pathogen. So clearly there is a very strong metabolic regulation of whether a particular species will become pathogenic or stay symbiotic in that kind of environment. And what we also see in some of our microbiome constituents is that if you have one particular constituent at a very high level compared to the others, then it starts behaving in an antagonistic manner rather than being symbiont.
The last question probably asked by Binoj. How does such knowledge in rhizobiome change the future for biofertilizer production?
I guess in a way it makes it more specific and more tailored to what is needed. And it definitely helps to minimize the footprint for fertilizers, for example. And that's exactly what our main aim is also. And what we are also doing at the same time is to see whether we can also minimize the footprint for not just the fertilizers, but also for water. So we are looking at trying to incorporate these microbiota in actually produced, like for example, there's a hydrogel that would just incorporate these microbiota to reduce both the fertilizers as well as the water footprint.
Thank you very much, Professor Naqvi. We are completing this session here, and thank you for joining with us, giving the questions of all our participants. And I hope we will be in touch with you in future also. All the participants are requested to follow the feedback link, which will be activated tomorrow after 2 p.m. And we will be back after a short break by 12 o'clock with the lecture by Dr. Deepak Manda. Thank you, everyone.
Thank you.